Hi, I'm Glenn Yap, and welcome to the Xray crystallography laboratory. Um, what do we do here? Well, if you have a crystal, and let's pretend that these are atoms or molecules, then what you have is actually a pattern of electronic density. So, if you have a pattern like this, and then you hit it with electromagnetic radiation, which are waves, then what happens is that you're going to have interference in the crystal, right? And the resulting waves that are coming out of your crystal will either be constructively interfering with each other, in which case you'll have a reinforcement of the wave front, or they will cancel each other. So if that's what we're doing, then we don't have a pattern called a diffraction pattern. So basically, what we are doing in this whole lab is to get the diffraction pattern of crystals, and by carefully measuring the spots, the diffraction pattern spots, their intensity and position, we can then figure out what was in the crystal that was causing this. And of course, you know that it would be the molecule or the arrangement of atoms in the crystal that would be doing this. Or, in other words, the crystal structure. Now, over here, we have a very intense X-ray beam source right there. And it's monoclinated, it's going through here, it's interacting with the crystal right at the very tip of that mount right there. And this is the camera. This is the camera that actually measures the diffraction pattern. And in the old days, what we do is we do a serial detection, wherein you have to go and drive from point to point and measure that spot. Then later on, this was improved, the technology was improved, and we were taking snapshots. This unit detector does it in a movie fashion. So the net result is that we're getting data at a much faster clip. Back in the day, for instance, it wouldn't be surprising to do a small molecule structure in like 4 or 5 days. Now, we could put in a crystal, and in 4 or 5 hours, we could have a crystal structure. Obviously, if we are screening proteins, proteins are very big molecules, it will take a longer time. This diffractometer, for instance, was purchased specifically to screen protein crystals. Now over here, we have another diffractometer, and it has this detector here. This is a new upgrade of our system. But I'd like to show you that this one has two sources. You have a copper source here and an X-ray tube using molybdenum in the back. Now, why is this such a good thing? Well, because of the physics, if you have a lower wavelength X-ray source, then that will tend to be more, what's the term? It's easier to get fuller coverage of all the data from your diffraction pattern. It's also less absorbed by your samples. If you have a crystal that is shaped like a needle, then this would be better. If you have a longer wavelength X-ray source, then it will tend to be absorbed by the crystal a lot more, which might be a good thing and a bad thing. Bad thing would be that we might not be able to see as much spots. But the good thing is that there's a certain thing called an anomalous dispersion And that allows us actually to figure out if your crystal is left-handed or right-handed. So these two diffractometers are actually quite productive. And just to give an example, before this whole COVID-19 problem that has slowed things down, we sort of like on average get a paper once every month. You can see that this thing is still not quite done here because upgrade is actually ongoing. Hopefully within this week this will be finished.
Do you run crystals for students, or do students run their own samples?
It depends on the student. If the student wants to run their own samples, they can. They'll have to undergo a training session with me, of course. If you don't want to do that, that's also fine. You can submit the samples to me. Just fill in a form, and then I will run it.
And for students who want to learn more about crystallography, you teach a course every spring?
Yes, I do. It's the Inorganic Special Topics Introduction to Xray Crystallography, and that's a 3-credit graduate